Hi again, and welcome to the fifth lesson in the iCow soil biology course, specifically designed for smallholder farmers who are interested in learning about the biology in their soils. To refresh our memory, we're drawn back to the soil food web picture again, this picture that was designed by the infamous Elaine Ingham from the Soil Food Web Foundation. We've learned that the soil food web follows the same principles of most food webs, where each organism or sets of organisms play a functioning role in the soils that contribute to the overall soil ecosystem. We also learned that in the soil food web there are organisms that we as farmers consider to be less than desirable. However, in the big picture, they play critical roles. We understand that plant succession above the ground is dependent on the trophic levels within the soil below them, and that the first trophic levels are the photosynthesizers upon which all living creatures depend in some form or format. The second level are the decomposing mutualists, pathogens, parasites, and root feeders. In this group, we learned about the bacteria and the fungi, and we learned that bacteria are the smallest of the organisms, some being as small as 1 micron, and that fungi are hugely beneficial, playing a critical role not only as decomposers, ensuring that organic matter is decomposed and returned to its basic nutrient components, plant-soluble nutrients. They also act as the cement binding micro and macro soil aggregates, allowing for improved soil integrity and structure, improved air and waterways in the soil, and they also act as a neural network capable of sending information between plants across thousands of acres. This helps plants react to pests, predators, toxins, and other threats. Today's lesson is on the third trophic level, the shredders, grazers, and the predators. And we shall be learning specifically about the protozoas. So, protozoa are a group of unicellular organisms, also known as single-celled organisms. They have no eyes or features that we think are necessary to get around. Instead, they use impulses, other senses, some that we don't even know, and some structural advantages to get around in their world in the soil. They're typically very small and only able to be seen under a microscope, but they're several times larger than bacteria, which we know are as small as 1 micron in size. Protozoa typically reproduce through what is called binary fission, which is by simply producing a replicate of itself. When conditions are favorable, they can reproduce within 8 hours. Though it'll take a lot longer if conditions are unfavorable, like lack of food, sudden temperature changes, waterlogging, etc. In this picture, you can see one testate amoeba undergoing binary fission, creating an exact replica of itself. Protozoa eat bacteria. Each protozoa can eat up to 10,000 bacteria every day, and they excrete soluble nutrients into the soil solution. When bacteria levels rise, protozoa numbers begin to rise too, as their food, the bacteria, are in abundance. And soon they'll eat so many bacteria, that the bacterial biomass reduces. And when this happens, and there are not enough bacteria to feed the growing population of protozoa, they'll typically encyst. This means that they'll go into a dormant stage where they're not using energy and they're not eating, and they can be seen under the microscope as round circular structures with double walls. When this happens, if there is enough organic matter or plant exudates for the bacteria to consume, their numbers will build up again, creating conditions that will trigger the dormant protozoa to come out of dormancy, and so the cycle continues until a level of balance is reached where both populations are balanced. In this picture, you can see on the left there an amoeba, a testate amoeba. It's eating bacteria. And the picture on the right, you can see at the top a testate amoeba that seems to be breaking up, releasing its bacteria and those nutrients in the bacteria back into the solution. Below it, there is a encysted testate amoeba. You can see it as a round circle. And different types of protozoa. We have flagellates, ciliates, and amoeba, and they all look slightly different when they're encysted. We learn to identify them by whether they have double or single walls, and also the sizes of them. But typically they all insist into a round circle that we see in the microscope. There's another picture of a protozoan cyst. It's interesting. It is in our interest as farmers that the nutrient cycle taking place by the protozoa consuming the bacteria is constant and balanced in the soil. And we can enhance this balance by reducing disturbance to the soil and making sure the bacteria have enough food through plant exudates from our plant covers or through adding organic matter to our soils. And now we can see another reason why it's so important to keep our soils covered and undisturbed with growing plants. We benefit immensely from protozoa nutrient cycling by growing food and we benefit immensely from protozoa nutrient cycling by growing food and cash crops for ourselves and a constant cover crop letting the sun feed the microbes in the soil to keep the nutrient cycling happening and reduce our cost of production whilst building our soil health, and also really reducing the hard work that we've been putting into agriculture by digging our soils, which now we know is wrong. Protozoa don't just live a happy life of consuming bacteria and going into dormancy. They get eaten too. In the soil food web, they're eaten by micro and macroarthropods like nematodes and earthworms, and they're also eaten when in their dormant stage. Maybe some organisms see and eat them the same way we see and eat eggs. Last week, we learned that fungi also exist in spores and cysts and can last for thousands of years waiting for the right condition before they wake up. Protozoa, however, protozoa cysts don't last that long. They rarely manage to last in a dormant stage for more than 9 months. They're far more delicate and fragile. They do need to exist, though, in moist conditions, and that's why it's really important that if we are following rain-fed irrigation, we don't leave our soils bare. We leave our soils with growing plants, even if it's weeds and grasses and anything wild that might be growing in between when we're cropping, just so that those plants are photosynthesizing, still feeding the microorganisms below the ground and making sure that they stay fairly moist as well, because the microclimate will be slightly different. In the poop loop picture we can see here, starting from living plants that decay into dead organic matter, they're eaten by the first trophic, broken down by the second trophic level, the fungi and the bacteria, where the nutrient that they have absorbed is now accumulated in the bodies of these organisms. And then when they're eaten by the protozoa, the amoeba, the flagellates and the ciliates, they actually change the nutrient composition from nitrate to ammonium. So again, protozoa are critical in being able to move our soils from that succession level, early succession level, further up to enable us to grow higher succession level plants like grasses and shrubs, herbs, and onto trees, fruit trees, and other trees. Without them, our soils get stuck in the early successive stage where we're only really able to produce weeds. What's really cool is that even as these tiny guys are what is really cool is that even as tiny as these guys are, they're selective about what they eat, and the micro and macroarthropods that consume them are also selective and will spit out protozoa that they don't like, or actively seek out ones that they do. The protozoa do the same with bacteria. But why do we find this so strange and exciting? We humans also have our preferences of foods when given a choice. What we do know is that we don't know a lot about the soil food web dietary choices between organisms. And here is another picture just showing, and I'll read it straight off, that soil nutrients generally occur in two forms: inorganic compounds dissolved in water or attached to minerals, and organic compounds, part of living organisms and dead organic matter. Bacteria, fungi, nematodes, protozoa, and arthropods are always transforming nutrients between these two forms. When they consume inorganic compounds to construct cells, enzymes, and other organic compounds needed to grow, they're said to be immobilizing nutrients. They're building them into their bodies, and they are no longer available in that form to anything else. They're immobilized. But when these organisms excrete the inorganic waste compounds, they're said to be mineralizing nutrients. And that's what's exciting, and that's when they're really useful, and we see the usefulness to us as farmers, as regenerators. There are several types of protozoa in the soil which fall into three main groups: the flagellates, amoeba, and ciliates. And like bacteria and fungi, there are some that prefer aerobic or anaerobic conditions. Just to remind us, aerobic means conditions where oxygen levels are 6 micrograms per gram and more. And anaerobic is where oxygen is lacking or below 4 micrograms per gram. And we know these conditions change when soils become stagnant with water or are heavily compacted where airways are damaged. Knowing and seeing the different organisms in our samples tells us a story at a glance, whether our sample has come from an area that is aerobic or anaerobic. And isn't that so cool that we can actually identify the conditions of the oxygen within our soils through soil life? So most protozoa are motile. That means that they can move. It means that they can hunt and they can search for food, and some even hunt in packs. Some move very, very slowly though. Some have the ability to produce funny leg-like things called pseudopods. These are non-structured and change in shape and direction and are made up of the cytoplasm, which is the internal jelly stuff like in protozoa. Typically, amoeba move this way, and you can see the top picture of the amoeba. Basically, what happens is as it senses the direction it's going to go, it oozes in that direction. A pseudopod comes out and just literally just directs the amoeba the direction it's going. And it's quite interesting to watch under the microscope. I've only actually seen one in all the samples I've looked through. I've only seen one naked amoeba. Many amoeba don't do the same thing. They do have their little pseudopod legs that come out of the bottom picture. There is a testate amoeba, a little bit like a hermit crab in what we know here. So they have a shell around them, and the amoeba lives inside the shell. And when conditions are right, it puts its little arms out of the shell and it pulls in the bacteria and that it's feeding on. They move very, very slowly too. Sometimes a naked amoeba can actually move quite fast. They look like mercury spilling through a sample. Very cool. Another mode of motion is by the use of flagella, which are one or more very thin hair-like tail structures some protozoa use to create motion. They can use their flagella to go forwards or backwards like a little propeller, and we call these protozoa flagellates. You can see on the picture on the right there. And finally, some have tiny hairs covered all over their body, sometimes not all over, sometimes in parts, and these little hairs are called cilia. These hairs or cilia move in patterns that create motion and enable the protozoa to move quickly, quite quickly, giving them the name ciliates. And the ciliates are the biggest of the protozoa. They actually, on a slide, they go zipping and whipping around. They look like little fleas under the microscope. Whereas the flagella, because of the way they move, they bumble along. Of the protozoa, the amoeba move the slowest, although sometimes the naked amoeba move quite quickly. The flagellates move fairly fast in a bumbling and almost unbalanced pattern, and the ciliates move the quickest. And as I've said, they're the biggest of the group, while the flagellates are the smallest. The ciliates are also our anaerobic indicators, as they come out of dormancy and reproduce when conditions go anaerobic. Again, really, really interesting to see when we see these three groups encysted. The smallest cysts are the flagella. The largest cysts are either the amoeba or the ciliates. And we can see from the cysts, some of them have a single layer like the flagella and the ciliates, but the amoeba have a double layer, so it's like a double ring when we see them under the microscope. So let's have a look at some of the pictures of the three groups. Here are a picture of some of the flagellates. You can see some of them have a flagella on one end, some of them have a flagella on both ends. Some of them actually use their flagella to anchor themselves whilst they're feeding. Very cool to watch. Here are some amoeba. Up on the top left is a beautiful golden testate amoeba called an arcella. The picture on the bottom left is a naked amoeba, and you can see inside its cytoplasm, you can actually see the bacteria in there. It's harder to see the testate amoeba because of the shell that's covering it. In the picture in the middle, if you look really, really closely, all of those red dots are by amoeba. And there were, in this one slide picture, and I think it was either a compost or a vermiculture, there were over 30 amoeba. That shows us that the sample we were looking at had plenty of food to produce so many amoeba, which meant that there were plenty of bacteria. It could have been a sample of compost that was actually still going through its decomposition cycle, where these things were all breeding in such high numbers. It could have been from a sample where actually the compost was warm. I don't remember where the picture was taken from, but that's what I would imagine. The one up on the top right again is a picture of a testate amoeba. Very beautiful. Some of them are ornate and really beautiful colors. Don't they look like jewels? The gold one, the purples, and they all have such beautiful patterns on them. And then below is a picture of a testate amoeba. And this, because of the ornate second ring on the outside, we know that it was, sorry, it's an encysted amoeba. We know that it was an encysted testate amoeba. Here a picture of some ciliates. The one on the top left was taken from the bioslurry sample, where these ciliates just look like whales moving through the sample very slowly, just hoovering up the bacteria in their wake. So beautiful and graceful to watch. The one in the middle is a stalked ciliate, so it anchors itself onto it's anchored itself onto a little bit of material there. The picture on the bottom right was actually the same stalked ciliate. When it opened up, and out of the little vacuole at the top of it, it had these little spinning cilia that were creating a vortex in the liquid around that was sucking in bacteria into its body. And because it does this, it's actually called a vorticella, which is such a beautiful name. They're so pretty to watch, and they normally come out in little clusters, and they look like little the little things we see in the sea, the little anemones. They look like that in the sample, all out there waving around, moving on their little stalks. And then, interestingly enough, if there's any commotion, any sound or vibrations, or loud vibrations, I noticed when banging the table that I was looking in the sample through, that the ciliate quickly retracted on its stalk. It sprung back and closed itself in to protect itself. And then the other two pictures, the bottom left and the top right, are pictures of typical ciliates that you can see with their cilia, all of their hairs. Now let's see if we can actually look at some under the microscope. And now we know about the protozoa. Next week we'll be learning about nematodes. Don't forget to tune in for lesson number 6 on nematodes. I don't know what this ciliate is called, but it looks like a Viking ship to me, so I'm going to call it Viking ship ciliate. It's moving around, harvesting up in the front of its face there. It can move backwards and forwards. It's just harvesting away, nudging things as it goes. Really like a big ship. These are some of the biggest ciliates that I've seen. On the screen where I was looking at it on the computer, it's likely to be about 120, 130 microns long. Here's another ciliate in bioslurry, and you'll see it moves like a Viking ship around the little aggregates of material in the bioslurry as it harvests up all the bacteria that it can find. It doesn't bump into things, it seems to swim around things, over things and under things. So here are a whole bunch of ciliates. They're around a bit of an aggregate. Some of them moving very quickly. See, they're like little fleas. They move so fast. And then you can see some tiny weeny little things under there that are moving around in the left-hand corner. And those would be the flagellates, and you can see the difference in movement. You can also see the difference in the size. I can't focus on all of the ciliates, but you can see some of them that stop and that are in the upper levels of the slide. You can see the hairs on them and the way that they move. The others a little bit deeper down. They have quite a depth that they can swim in this slide. But there you go, these are the ciliates. Again, defining very anaerobic conditions, and these are taken from a bioslurry sample. So here's going to be a video through vermiculture, and in the vermiculture here you can see very, very high levels of amoeba and many different types of amoeba. There are big ones, there are small ones. You can see there are some that are going through binary fission. There are gold ones, there are clear ones. So great diversity, and we know that the amoeba are the protozoa that are very, very functional in changing the nitrites that they're getting from the bacteria into ammonia for your plants, allowing you to have higher succession plants growing in your soil. And you can see we're seeing some fulvic acid there. That's the brown stuff, and like the aggregate. The browner it is, the darker it is, it's humic acid. So there's some dark humic acid there. The browner it is, it's fulvic acid. And you can just see through the vermiculture the various different prosits that are in there. Well, not different prosits, it's all the same, they all seem to be amoeba. It is aerobic, so we're not seeing ciliates. It's not excessively wet, so we're not seeing flagellates. Those would require more humidity and moisture.